immune system can remember that problem or some disease or something come into the body. So this is how the science often happens. So then in 2002, the Johnson, first article is delivered from the Dr. Johnson, the identification of genes that are associated with the DNA repeats in the prokaryotes. So there we can see this by direct repeats varying in size from 21 to 37 base pair here, the direct repeats. And we have a spaces. So this is the direct.
I'm requesting all to unmute.
Prokaryotes. So there we can see this. Hello.
Yeah.
Salim, go forward. Hello. Dr. Busman Singh. Hello. Salim, move forward. Hello. Is it okay?
Now it is okay, I think. There was YouTube live that also started in the echo sound.
Okay.
So we have stopped that, and I request all other participants also to keep yourself muted, please. So I am muting all and unmuting. Yeah, I think it is now it is okay, I think. There was YouTube live that also started in the echo sound.
Okay.
We have stopped that, and I request all other participants also to keep yourself muted, please. So I am muting all and unmuting. Yeah, I think it is now it is okay, I think. There was YouTube live that also started in the echo sound. Okay. We have stopped that, and I request all other participants also to keep yourself muted, please. So I am muting all and unmuting.
Okay. Salim, start. Hello. Hello.
Yeah, please continue. Now it is okay. Thank you.
Okay. Salim.
Yeah, please continue. Yes.
Hello.
Yeah, yes, please. You can continue. Hello. Yes, you can continue now.
Hello.
Okay. Please continue. Now it is okay. Thank you.
Okay. Salim.
Yeah, please continue. Yes.
Okay. Thank you. Thank you. Sorry for the disturbance. And so we'll move on to that. So this family characterized by the direct repeats. So that I said varying in size from the 21 to 37 base pair. That is called as the interspaced by similarly sized non-repetitive sequences. So this refer to the family as the clustered regularly interspaced short polyentropy repeats CRISPR. So this is the direct that consists of the two different things, direct repeats and as well as spacer. I will show you the next slides. What are these things? So now move on to the CRISPR Cas19. So this is that unique technology that enables genetic and medical researchers to edit parts of the genome by removing, adding, or altering section of the DNA sequence. So this is the three important thing. So we can know, but removing, adding, altering section of the DNA sequence is very, very important. So where we have to insert how it will be by using the case 19, case 9, the protein. So it has a lot of RNA guided sequence also there. I'll show you the upcoming slides. So these are the most rapidly evolving technology which has the revolutionary biological research. So it represents a family of DNA repeats in most archaeological, some of the bacteria and bacterial 40% genome. So these, the particular removing, adding or altering so provides the acute immunity and resistant to the foreign genetic element such as virus and patches. So what makes CRISPR system the ideal technology? Why we want to use this, CRISPR as an ideal technology? So we can see the key in enabling activists to the become next big classes. So we have this CRISPR Cas19 as a high potency. That means removing, adding, and insertion. That is called as the cleavage efficiency, and it has the more specificity where we have to cut and insert, and we can add into the new DNA. So that the broad applicability both, even we can use as a in vivo and in vitro applications and a simple editing tools. So we have a guide RNA plus protein. I will show you the next 1 or 2 slides later, and allow unprecedented ability to scale and optimize at speed. So and then we have a one-time curative treatment. So once we can identify the particular gene, so where we have to cut or insert and delete the particular gene place, and then it has that potential one-time curative treatment, and we can see that the broadest potential to modulate the genes, the ability to address any site in the genome or foreign genomes. So ability to target multiple DNA set at a moment, simultaneously, and it has the multifunctional programmability. Even that's what I said, the delete, insertion, or the repair genes. So the genome manipulation by the CRISPR is a remarkably flexible tool for the genome manipulation. And the genomic manipulation, it requires that 3 different mechanisms. One is a Cas9 protein, another one is the engineered small guide RNA, that is a gRNA, and it requires the DNA template sequence, the target sequence. So we'll see the, so here that the system CRISPR Cas19 consists of 3 parts. One is the Cas genes. Here there are 4 to 20 different Cas genes, and CRISPR area that I already showed about that repeat and spaces DNA in the between the CRISPR area, and the leader part. This is contains a lot of adenine and thymine rich, and this is located upstream side of the CRISPR array. So that already as discussed about how this CRISPR can useful for the how it will work on the some of the application. We can go for the gene disruption. We can delete and gene corrected and insertion, and we can go for the some deletion by using the homology directed place. We will see what are these things. So now it's a Cas9 protein. It's a CRISPR associated protein. It's also known as molecular scissors. So this is the particular associated CRISPR associated protein has that molecular scissor, and it has the RNA guided DNA endonuclease enzyme. And this particular Cas9 associated with the adaptive immune system in the particular protein. The genes, the Streptococcus pyrogenes. So this Cas9 is isolated from the particular Streptococcus pyrogenes. So here that structure of the Cas9 structure. So we can have there is a multi-domain structure. We have a guided RNA, and there is a cleavage site and PAM. This is very important. It's a protosphere adjacent motif. It's a 2,6 base pair DNA sequence immediately following the DNA sequence targeted by the Cas9 nucleus in the CRISPR bacterial adaptive immune system. So here Cap9 contains a 2 nucleus. So here we are saying that one attractive target for such study is the Streptococcus pyrogenes. That's what I showed the previous slide. Cas9 the prototypical RNA-guided DNA nuclease used for the genome editing. So the Cas9 is an excellent model protein for the comprehensive deletion study because of the multi-domain architecture. That's what I said, multi-domain architecture. Sorry. Sorry. And availability of high throughput assay for either DNA cutting or binding. So that's what it has, the multi-domain structure. We have the guided RNA, and we have a PAM, and we have the darker DNA double helix standard. So, and even functionally, so these Cas9 targets and cleaves the DNA in multi-step process. So first of all, it's a Cas9 nucleotide molecule forms a complex with the guide RNA, that gsRNA, so containing the 19 to 22 base pair variable spacer, variable spacer here. Okay, sequence that is complementary to a DNA target locus. The primed ribonucleoprotein RNP complex that surveils genomic DNA for a protopressor adjacent PAM. Okay, this place. Protopressor adjacent motif 5 NCGS. In the case of Cas19, when N is any nucleobase, that initiate the transient interaction with the protein to search for an adjacent 26 base pair target sequence. So here, adjacent 26 base pair. So the single guide RNA directs a Cas19 endonuclease to introduce the double stranded here, to double stranded break at a specific site in the genome, So the endogenous RNA repair mechanism later repair the genome using the two different mechanisms, namely the predominant error-prone, non-homologous end joining, and less frequent homology derived repair, HDR. Okay, HDR, which requires the donor template. So this requires the donor template, and non-homologous end joining has been used to disrupt the genome sequence to create the deletion and insertion. While HDR, the homologous directed repair mechanism, has been used to alter the specific region of the genome editing exogenous repair templates. So in addition to this, modified version of the Cas19 protein, catalytically dead Cas19 has been utilized to either activate or repress the target genes, These mechanisms are known as Crispr activation and Crispr interference respectively. So if your target is present, that DNA is present, the double stranded DNA helix unwound, it's unwound, allowing the gRNA, the guided RNA to anneal to the DNA, and from a stable RNA DNA hybrid structure called R loop. So here is this, this is the R loop, and the formation of a complete 20 base pair R loop region. Here this triggers the conformational change in the Cas19 to form the catalytic active complex. So therefore we have the Cas19 contains the two nucleus protein. One is the H&H domain. So already is the H&H domain, and we have a RUC-like domain here. So these cleaves the non-complementary states. The H&H domain can cleave the complementary state to the cRNA RNA guide sequence. So the Cas9 protein induce the double stranded breaks at the target site. So the Cas19 has a bilobed architecture consisting of RE recognition lobe responsible for recognizing the binding DNA sequence called under the nucleus lobe, which passes the H&H domain and REC domain that cut the target and non-target strands of DNA respectively. Cas9 is postulated to have evolved domain obtained from a progenitor RUC domain. Significantly, the Cas9 orthologous possesses the manifold, multifold structure. So the function of REC2 here, the function of the REC2 is ambiguous, but in the thought to act as a conformational choice to trigger the DNA cleavage. So raising this question of how he has accomplished the effect. Thus the multi-domain and the multifunctional nature of the Cas9 makes them an excellent model system for exploring the domain deletions. So Cas9 large size also there's some other limitation for the by using the Cas9. So because of the large size also complicate in delivery using the viral vector. So therefore that knowledge of functional deletion may thus facilitate the delivery of genome editing therapeutics. So here this slide shows the basic flow of the CRISPR genome editing system. So here I am using the some of the application of CRISPR Cas9 in the plant system. So here we have an engineered CRISPR Cas9 system consists of two components, the Cas9 here the Cas9 endonuclease and a single guided RNA. Single guided RNA. The single guided RNA contains a baser sequence here. Followed by the 79 nucleotide of an artificially visited tracer RNA, artificially visited tracer RNA and cRNA sequences. So the spacer sequence here, the spacer sequence is typically 20 nucleotide length and specifically binds to the target site. Okay, for target site and containing the 5' NGG to 3' PAM site to the PAM site. And at the 3' end, so which is highly specific for the gene of interest. So once the piece to transactivating cRNA and cRNA sequence forms the stem loop. This is the stem loop RNA structure that binds to the Cas9 enzyme. So tracer RNA hybridase and joins Cas9. So assembly of single guided RNA attached to the with the target sequence called and the Cas9 vector construct to the the transformation of the vector constructed into rice via the different transformation techniques, and then we can go for the after construct and transfer with the vector of the particular gene. So I mean Cas9 and other guided organ and the target site. So we can go for the screening and selection of the rice mutant plant based on the phenotypic change, based on the phenotypic changing, and the restriction enzyme that lose generation CRISPR-Cas9 mutagen plant line. And so we can use a lot of assay to identify the endonuclease utilized in the surveyor assay. So next, and then we can use the next generating sequence also. So feature analysis, feature analysis to obtain the tDNA free plants, and further experiment to prove the phenotypic changes caused by the knockout of the gene under investigation. So here I'm giving another important disease called hemophilia. So by how to we can use this CRISPR-Cas9 to great potential to correct the disease causing the mutation in the hemophilia mice. So hemophilia is an monogenic disease with the blood clotting factor 8 deficiency caused by the mutation in the factor 8 gene. The current and emerging treatment such as 8th protein injection and gene therapies we have delivered transgene in ribosomes are costly and non-permanent. So therefore that we are using the CRISPR-Cas9 based in vivo genome editing method. So combined with non-homologous engineering enabling permanent chromosomal integration of the modified human B domain deleted FB at the albumin locus in the liver cells. So therefore, based on this finding, this genome editing approach may affect on the efficacious long-term and a safe treatment for patients with hemophilia. So then move on to the next one, that application in the cancer therapy. So here the Cas9 nucleus directed to the target DNA by complementary base pairing with its bound guide RNA in which the target site is followed by the PAM sequence. So that's what NAG and NAG. that the right hand side we can see that already I mentioned about that we can use the CRISPR-Cas19 in the multiple target site. So here the two guide RNAs are targeting to different sites A and B. So can be used to simultaneously to remove the longest stretch of the DNA, and a new DNA can be inserted using the donor DNA using homologous direct receptor. So here we have a lot of application by using the CRISPR-Cas19. We can see that the human gene therapy already I showed about that hemophilia B. So how to change that in a particular gene, and we can use the agricultural crops. I already shown in the plant material also, and we can use this CRISPR-Cas9 application in the synthetic biology, and we can go for the programmable RNA targeting, and we can use the drug target ID. And we can go for some of the ecological vector control, say for example, mosquitoes, deviation, and etc. And then again the viral gene disruption, pathogen and gene disruption. So the CRISPR-Cas technology has evolved rapidly and has revolutionary the scientific field from basic research to the clinical applications. Undoubtedly, this technology has provided many promising avenues for a greater understanding of various diseases and their treatments. So innovation in the CRISPR technology are likely to increase its precision, effectiveness, and safely for its future preclinical and clinical application in different metabolic disorders. In the next decade or so, this technology will play a pivotal role in identifying the new drug targets and developing the dependable therapeutic strategy for several diseases. Thank you. I am ready to take the questions.
Thank you, Dr. Rajagopal, for this very enlightening and useful talk. I think this would be, it is actually this type of talk that we wanted to host in this series, because a lot of our participants are from masters or MTech or BTech degree. Yeah. So if there are any questions, participant can, there is one question regarding which part of the CRISPR is variable so that it can be used to cut DNA at any site.
Actually, it's not working on the CRISPR. There is a, I already mentioned about that, repeating site, especially in the Cas9, the protein, associative protein can do the endonuclease, right? So that can be bound with the particular target site, the DNA. So that can be cleaved. So that's why I already showed the last slide. So it can work on the multiple target site, A and B. Okay. So that's what the answer for this one.
So basically, there is the specificity comes from the guide RNA.
Yeah, gRNA. Yes, gsRNA. Guided. Yeah.
Okay. So yeah, thank you very much for this talk and for your answers to the questions. If there are any other questions, you can please send by email or you can type here. We would like to begin for the second talk, because right now there is another talk. There is just one more question. How can we identify that this gene is defective and is causing the disease? That's the question from.
So actually, it's a Divagar Misra. It's a good question. Actually, we can use a lot of techniques to identify the genes. See, for example, that's what I showed that hemophilia B, the gene, the BAD genes, right? So based on that molecular technology, how can we can use a lot of identify the gene first. Then we can, especially in the factor also for the particular gene can be we can insert, delete, or we can remove the particular gene. Thank you. Is it okay?
Yeah. Thank you. So with that, I thank you, Dr. Rajagopal, and I request Dr. Prakasha to share his screen to start the second talk.
So thank you, Dr. Pushpendra Singh, to invite me, and once for thank you to participant also. Thank you so much.
Thank you very much for your time, and this was really very helpful. And I have introduced Dr. Prakasha briefly. He has done his PhD from University of Göttingen in Germany, in 2006, and since then he has worked in many places, in University of New Mexico, and currently he is at Mayo Clinic in Florida. So I welcome Dr. Prakasha, and his slides are already on your screen. So please go ahead, Dr. Prakasha.
Oh, thank you, Pushpender Singh. So thanks for inviting, and it's a pleasure presenting some of my previous work. So in fact, currently we work on therapeutics, drug screening. So this is a part of the project or the proposal. We worked almost for 10 years, and until 2018. So it's not the current work. Current work is, I'm not presenting the current work, but this is like a snapshot of what or the techniques that we have used in order to investigate or unravel some of the host genetic factors. I hope you are all able to see these slides. So let me go over. I'm glad that Senthilkumar and previous speakers have given some of the intro to the RNA Seq and Genome-Wide association, or some of the techniques of the genomic tools. Here, let me see if I can go over. The primary goal of this study was to understand or unravel the host genetic factors prevailed or responsible for the complex disease outcome. So one of the complex disease we selected here is the malaria, because it's not a monogenic disease. It is let me pull this out. I have a lot of okay. Okay, so because it's some of this one is blocking. So I'm sure you have all known that the whole genome, our body is composed of more than 3 billion DNA, that's the single nucleotides, and they are all stored in 23 chromosome pairs. And so far, the science from a lot of studies and whole genome sequencing, it has been found that around 21,000 genes are present in the human. Basically, they are coding genes. Those genes are coding for the protein. Rest that comprises only about 1.5% of our genome. The rest of the 98.5%. So as you know, it's all coded. It's all coding for it's related to non-coding RNA, as well as pseudogenes, some of the regulatory sequences, as well as transposons, and most will be a junk DNA. So which we don't know the role or the characteristic of the part of the genome. And here, so in order to understand the complex or complex diseases that are caused by a combination of environment as well as the genes, it's very hard to understand or pinpoint what genes are responsible for causing our severity, or for example, the cardiovascular disease and cancers, you name it. So including the infectious disease as well. It's not a single or a monogenic factor. So not a single gene. So although even the reason why I'm emphasizing the complexity is even though all humans are made with made up of same nucleotide sequence or same genes, but only we vary with individually between two individuals we have a 0.1% genetic change. It's because it's mutations. So those 0.1% can make you sick or resistant or protective from the diseases. So primarily the goal of this proposal or the complex understanding the complex disease is to understand what those 0.1% difference between the two individuals, so we can therapeutically or so it can be used for many downstream applications. So let me go to the next slide. So with this small introduction, let me move on to the disease that I want to present. So it's malaria. So malaria, you all know that it's very complex disease, and governed by a single factor. So although the main criteria is to get bitten by mosquito carrying the parasite, but the disease outcome is primarily driven by the host genetic factors. So you all know the 90% of the death occurs in Africa, and India also, there is still ongoing malaria mortality, but mostly the primary the impact is found in Africa. So the malaria is caused by primarily 5 parasites. Those are very lethal. Falciparum, that's the primary very lethal one, followed by vivax, that's mostly found in India and some of the Asian countries. Malaria and oval, and these 2 are in South America. And recently there is one of the parasite that is transmitted, transferred from monkey to human is an Rna Seq in primarily in the Asian region. Mekong primary, and also we recently found, if you see in the publication in the bottom, we recently found one of the parasite from monkey to human. It's a Brazilian, Amazon forest. That's also known to infect the humans. So there's a lot of generation of there's due to change in the habitat and the afforestation, there's a exchange of parasites between human. It's like basically they can find different hosts. So with that, let me move on to the next slide. Okay. So before I begin exploring the genomic techniques, so what I want to highlight is so malaria parasite primarily eats or it uses the hemoglobin. So that's found on the RBC cells, red blood cells. So primarily the malaria parasite infect the RBCs, then eats up all the hemoglobin. So while digesting the hemoglobin, only the globin part is useful for the parasite, not the heme part. So heme part is primarily it is highly toxic to the parasite. So what it does is it will crystallize the heme part, and it's an iron actually. So it crystallizes the heme part, and it becomes the hemogen. It becomes like a rock, iron rock, and it cannot be excreted. And what it does is it will store this iron, the hemozoin. It's called as hemozoin in the epicoplast of the parasite. Although I'm not presenting all the background. So there's a component, there's an organelle within the parasite. It will store all the toxic materials in that in that component, in that organelle. So once the RBCs are lysed, so you might have seen the malaria cycles, and every 48 or 72 hours you see the febrile, the fever. So that's when the RBCs are lysed. Millions of RBC cells are lysed. That's when you get the shock, like a cold and chill and the febrile conditions. During that time, once the RBCs are lysed, so the macrophage, those are the phagocytes in your blood, they engulf, they eat up everything that's floating in the circulation. So once the macrophage are so you can see in the bottom, everything is taken up by the hemozoin is taken up by the phagocytic cells. So it will stay inside. The hemozoin stay inside. It cannot be excreted either with kidney or so it will stay within your body. So for example, you can see here in the left hand corner, this is spinal cord and the bone marrow. So it will be enriched. All your organelles are enriched with hemozoin, which is the immune modulator. It is not it cannot be used as a metabolite or anything, which what it causes your immune dysregulation. So your immune system is affected. That's when the patients or the individuals who are affected with the malaria dies, or in addition to the parasitic parasite, this is the additional cause of mortality or severity. So here I show this cycle of malaria, so once the mosquito bites, it injects the sporozoites. That these are the the parasite. The sporozoites within 2 minutes they will just go and reside in the liver. so the hepatocytic cells, they will stay for like 1 to 2 weeks. So you don't see any symptoms during the initial phase of the infection. Once they start multiplying, they will become murocytes. This is the another stage of the malaria parasite. So these are like sexual division, reproduction, and you can see thousands of them attacking the RBC cells. So they will infect the red blood cells, and this is when you start experiencing the symptoms, the chills and fever every 48 hours. So due to lysis, so you can experience the symptoms by many ways. So the parasite causes, and also you will get the treatment. So the symptoms can be the pathophysiological impact can be from drug or parasite-induced hemolysis. So parasite, the drug can also cause the hemolysis. For example, people who have the GAPDH deficiency, alpha thalassemia, and also like the sickle cell trait, they also get lysed by lysis of the cells by the drug. And another cause of the blood loss is the hemorrhage in, and also the other pathway is impaired erythropoiesis. So impaired erythropoiesis is the one that we are focusing in our team. So the impaired erythropoiesis means inefficient production of the red blood cells. So there is a lag due to the dysregulated immune response. There is a although there will be a lot of erythropoietin in the body, erythropoietin is the enzyme, the organ, the hormone that tells the bone marrow to make more red blood cells. So even in presence of a sufficient erythropoietin, you don't see production of enough RBCs. So the number of RBCs that are being killed by the parasite, the body cannot compensate. So you need to have certain number of red blood cells for a normal breath is normal the respiration, transfer of oxygen. So that's getting affected. That's when the mostly the children cannot gas, they cannot breathe. So either they have to be transfused with the new blood so that they can breathe, or they have to be like if they doesn't get rescued, they will succumb to malaria. So those are the two outcomes. So that's why when there is when there's a kid that comes to the hospital, they just transfuse, because your body is not able to recapitulate the required red blood cells. So this is just the study set we have been working for like many years. And it's an endemic area in Kenya. So it's a western part of the Kenya. It's called I think some of you might have heard in a Lake Victoria. It's the largest lake in Africa, and the water comes all the way from the Egypt, Nile River to it's the border of Tanzania and Kenya. It's very endemic. So every kid, you won't even believe. So we say because of the average more than 100 bites. So they get at least bitten by 300 mosquitoes. Okay. Among 300 mosquitoes, 100 mosquitoes, at least they will carry the parasite. So the malaria parasite. So once they bite, they will definitely get the parasite transfused, or the parasite in their bloodstream. So imagine, so out of 300 to the 365 days, 100 days, at least every day, if you are bitten by mosquitoes, you will get 100 days of malaria, but not all the time. An individual get malaria, because your host can fight fight off. So let me go and so little more information. So for example, so our primary goal was to identify why all kids doesn't get malaria, although they get bitten by mosquitoes, and we see the parasite in the bloodstream, but not all succumb, or not all get severe disease. That was the primary goal of this study. So here you can see on the left hand side, so all this kid, it has a milder, febrile. Like it's not febrile, it's not fever. it's a milder. It's the temperature, the fever is not more than 97 or 98. So it's a very normal, but still they have parasite. So another group of individuals who are like really sick, either they get cerebral malaria, which is like neurological sequelae, or they get distressed respiration, so they cannot breathe because they are anemic. So these two groups, we wanted to see what's the host genetic factors, or how the host is like fighting this parasite. Why there is a discrete outcomes. So what we did is, these are the characteristic of the two groups. This is like the individuals who are not severely sick. These are the characteristic. Another group is severely sick. So this primarily we divided them based on the hemoglobin level. So the hemoglobin level less than 5 gram per deciliter, and another group is more than 5 gram per deciliter. So that's the stratification criteria we use. So since we are this webinar is for the genomics, the application of genomic technologies. So what we use here is like we partitioned, or we stratified the individuals as sick and non-sick. Just remember that you don't have to remember the acronyms. So we grouped one group as a non-sick, but all of them have malaria parasite in their bloodstream. There is no difference in the number of parasites they carry. So there will be like irrespective of number of parasites, some are sick, some are not sick. So we group them and use 3 techniques here. So one is the in order to find out how their genetic factors are different. As I alluded in the first slide, 99% of the genetic material is 99.9. Actually, it's the same, but only 0.1% is different. So those 0.1%, what are the genes or genes and gene pathways are different between the sick and non-sick kids. Okay, so we use 3 techniques in order to unravel or to enumerate the genetic factors. So the first technique we used is GWAS, Genome-Wide Association Study. So here so we had like close to 5 million. We were able to identify millions of genetic variations. That is the single nucleotide polymorphisms, or even the VNTs, even the all so based on the nucleotide change, we were able to enumerate the changes in the whole genome. So the second technique we use is whole genome transcriptomics. It's called as microarray. So you might have already gotten the introduction background information on what the microarray means. And the other techniques, the latest technology is like RNA sequencing. So that's like the recent one. So you might know I can go a little bit of introduction, but I'll more primarily focus on their application, how they can be applied in the human genomics. So once we get all the varying factors, or the genes or gene pathways are identified, then we wanted to rank them. How likely would that they are implicated in this disease outcomes, the association. Okay, the genes, the SNPs, and the copy number variations. So a lot of information has been integrated using the softwares to identify the priority the targets. So those are the priority genes, high priority genes. So those, once you identify some of the genes or the pathway, then you can therapeutically target those individuals who are like severely affected or severely ill. Okay, so an example of this is like the outcome of the GWAS. So you can see all the dots are from single nucleotides. So once we see histogram, so once you analyze the likelihood of single the genotype, the nucleotide, and we also give a score of the p-value. So based on the p-value and several parameters, we rank them. So anything that is associated with more than 10 to the power of -3 or -4, you can based on the number of genes or number of nucleotides that spits from the generated from the jivas, you can rank them and select the ones that are most likely to be associated. And in the bottom panel, you can see we also map the whole genome in order to identify any of the genes or the part of the genome is affected, that is deleted or duplicated. It's called copy number variation. So for example, here the HLA-B gene in one or two individuals, I can show more in the next slide. So for example, this is like the sick kids. So sick kids, for example, just we took a snapshot of a single gene just to show how the HLA because it's a blood related, yeah. So in these two individuals, the whole region is deleted. So these two individuals are doesn't have this HLA copy at all. Okay. So you can, while the control individuals, not the control, actually the milder symptoms are non-severe anemia individuals have a normal copy. So an example, this could be playing some role. Okay, so only because it's affected in the sick kids, not in the normal kids. So this is one of the target genes, target pathway. So that's the outcome of the GWAS. So you can find the affected, affected SNPs, the nucleotide change, mutation, or in addition or in deletions, and also the copy number variation. You can enumerate, you can get the copy number data as well. Some of the copies are more, some of the copies are less. So in another two techniques that we use is microarray. So the difference between microarray and Rna Seq, although the outcome is slightly it is similar, but microarray, what it does is it will give you the differential gene expression levels. So whether the particular gene, the mRNA levels are high or low, you can also call as you can the fold change. You can also outcome can be presented as a fold change. But while the RNA sequence, what it does is it doesn't map the number. Actually, it will sequence everything that is present. All the RNAs it will going to sequence. Then you can annotate and find out the copy number as the differential expression level as well as the sequence. So if there is a change in all the gene, the mRNA can be present, it may not be expressed. So through RNA sequence, we can find out if there is a mutation or not expressed, it cannot be coded for the protein. So you can generate more data with RNA Seq, although it comes with expensive, the pricey. So the cost. The strategy that we used similar to the old genome Seq, the GWAS. So here also we stratified the subject as severe or non-severe. Then we use the CHIP and then hierarchical ranking and validation as well. So because once you find the genes that are affected, you need to validate. So here, for example, it's like it's like once you identify the number of genes, you can plot to identify whether it's like overexpressed. They are more overexpressed, more than certain number of copies. We call them as a fold change. We use, I think, twofold. More than the particular gene, if it is more than 2 and above, then they are red color. Okay, the black color spots are those are the genes that are not affected at all. So they remain same in between the sick and non-sick kid. The other ones are down. They are negative actually. So they are down regulated. So upregulated, it's another it's like similar to volcano plot. So upregulated, downregulated, and no difference. So then you take these genes that are affected and characterize them. Okay, so that's the goal of this, the analysis, the representation. So this is the heat map to show that, so for example, here sick kids are aligned in one side, the non-sick kids are aligned, based on the expression profiles. So you can see the pattern. Yeah, that means there is a real change in their the genetic component, the composition. Okay, so it's a different. And the RNA Seq. This is the RNA Seq. So you can, although we did the RNA Seq, only the outcome we were trying to exploit is the differential expression levels. So similar to the microRNA. So what happens in a year we use the equal number of individuals, and you can see here is the individuals who are normal have different levels of the expression pattern between some of the genes on the left hand side you can see. So it's called heat map. Okay. So once you identify once you compile pull all the differentially expressed genes or the SNPs, then you can actually study do pathway analysis. It's a network pathway analysis, or see if you just take all the genes that are affected, put into the metacore or some of the IPA gene go it will annotation. It will annotate and identify what pathways are primarily getting affected. Because this is a complex disease, it's not a single, it's not a monogenic disease, it's not a monolithic disease. So since it's caused by multiple factors, so you have to identify the pathways, not simple genes. So for example, this is an example of one of the pathway. It's a TLR pathway, and you can see here the genes that are affected are marked here. So these are the genes that are affected in this pathway. So then whether you can plan to modulate therapeutically all this pathway or in individual genes, because targeting individual genes require a lot of therapeutics, but you can modulate easily the single pathway. So it's an example. There's another one. It's a JAK-STAT pathway modulated by interferon alpha interferons. So an example, this is the publication we published. We showed that the heat shock proteins, for example, heat shock proteins and the downstream, the NF-kappa B, it's a transcription factor. All these are affected. So this is the pathway. It's affected because of the hemogen. As I said, the hemogen is the parasitic byproduct. We call it as a poop. Parasite because it doesn't use it. It will throw it out of the it's the system. And we were able to show that if there is a hemogen in the body, then it will trigger the inflammatory cascade through TLR pathway, and it affects the heat shock proteins, another lot of other downstream mediators, finally leading to erythropoiesis. You can see. So that's how the individuals who are affected, individuals who are loaded with hemogen, this parasitic product, will get impacted on the erythropoiesis. That erythropoiesis is the process of red blood cell production. So that pathway is affected. So whoever has inefficient erythropoiesis, they will have respiratory distress. They cannot breathe properly. So they will succumb to death. Either you intubate them. Intubate means you have seen the oxygen for the COVID. Similarly, you have to intubate or you have to transfuse them to rescue. So that's the only outcome. So let me go back. And another way, so this is another way of finding. You can feed all the genes that are affected or that were generated from the Jivas, transcriptomics, that is including microarray as well as the RNA Seq, and see what all the pathways that are affected. So it will be ranked. So you can, based on the rank likelihood of having more genes in this pathway, then you can explore in depth into what this pathway is playing role in this particular genes, in this particular disease. And for example, here you might some of you might know the genes that are affected, in this pathway. It's a network pathway. So it's a STAT3 and beta catenin, and these are the genes that were affected. You can just to get a idea of what all the pathways that are affected, what the role they are playing, how many genes are involved. This is the hemoglobin pathway. Yeah, so hemoglobin pathway encompasses hundreds of genes. So among hundreds of genes, even if an individual is affected by few genes, it will impact the whole pathway. So that's the idea of all this, CIMIC and ubiquitin pathway. So it's also affected in this malaria. So you can see a lot of genes are in fact they're upregulated actually. So they're actually not normal. So once you find some of the genes are then you have to validate. So sometime these techniques that we have used may give some errors. It's not accuracy. The accuracy levels are needs to be validated. What we do is we go and take a different set of population or individuals and isolate their RNA, and you can do qPCR or RT-PCR. Then you can quantitate. You can quantitate and see whether the what you found in those high throughput arrays are in fact it is true, or so that's how we did. And you can validate and identify the genes, and then you can once you confirm in fact they are affected, then you can plan for therapeutic applications. So this is the end of my slide. And overall, so this using all these arrays or the genomic techniques, we identified several genes that are actually impacting the transcription of genes, hematopoiesis, that is the erythropoiesis, and also signal transcription pathway. These are the three pathways that are affected in this malaria patients. And so now we have the pandora of the genes that needs to be therapeutically targeted. So next, that's what we are doing right now. We are doing a lot of therapeutics, immunotherapeutics, I would call it as. So you can target some of the genes, immunotherapy. You might have heard of immunotherapy. So we use the natural compounds, synthetic compounds, and already approved compounds and therapeutics to see whether we can modulate those genes and the pathways, as well as using as a RNA, like the ones that Pfizer and Moderna have developed, the delivering the RNA to recapitulate or as a substitution therapy, and also some of the therapeutics like glutamine supplements also we are using to see whether we can rescue or therapeutically. Yeah. Okay. Thank you very much. I think I'm up for taking any questions.
Thank you, Dr. Prakasha. So there is one question in the chat box from Jai Kishan Solanki. So this is regarding the ethnic variations that you have identified. He's basically asking whether the variation that you have discovered is only specifically present in African population, or it is present elsewhere in the world also.
Yeah, that's a good question. Yeah, that's true. So what based on these outcomes, we say it's only present in African population, because African population is there, like their genetic composition is different than ours. So I would say based on the outcomes, it's present in African population only, because the genetic variation is different in different races. Yeah.
Mm-hmm. Okay. So basically, but this also helps you identify the mechanism of the disease pathogenesis. So that is probably applicable for therapeutic applications in all the populations possibly.
Yes, generally, exactly. And also the studies have been done in India and in other parts of the world. So primarily, like the pathways will be same, but the individual gene variation will be a little different. Yeah. But that needs to be confirmed. So people have been confirming. So for example, like some of the hemoglobin genes have been affected in like closely related. For Indian continent is closely related to African continent. So there is likelihood that the genetic variations are identical, but I wouldn't say based on this current study they are all same. So it's very hard to say, because African populations are highly exposed to a lot of infections. So their linkage disequilibrium is different than what we see in the Indian population.
That basically need to be validated in other population to see. Okay.
Exactly. Yeah. Mm-hmm. And as you see in the malaria, so in Africa, you don't see the cerebral malaria. You see mostly severe anemia. So in Indian continent, we see a lot of cerebral malaria. So that's a neurological sequelae. Yeah.
Mm-hmm. There is another question about is it possible
to? There's a vaccine. Yeah, exactly. Yeah, the RTSS. Yeah. Go ahead, Pushpender.
So is it possible to turn off the reproduction genes of malaria carrier mosquitoes? So basically, he's asking if it is possible to turn off the genes, the reproduction related genes of malaria carrying mosquitoes.
Oh yeah, there is a lot of technologies now. They are transgenic mosquitoes, but it's not that easy. Yeah, it's not that easy. Parasites are so intelligent, and it's very hard to develop a vaccine or one single therapeutic to block it. It's very hard. So the vaccine that's been currently used in 3 countries, in Zambia, Kenya, and Ghana, it's the efficacy is 33%. Still, it's better, because we don't have anything. Something is better than nothing. So that's that target the surface antigen, but it needs a lot of boosters. So it is, the climate is it's pretty good, in protecting. If you are given a lot of boosters, and continuous monitoring is required, and also it's in the pediatric. So because once you get, I have been in Africa, and you see the severity of the disease, but sitting outside we don't see the severity. It's the people, once the kids come, because the kids come after once they see the symptoms, so they already under the respiratory distress and anemic. So there's no way to rescue the kid unless you have the transfusion. Even for transfusion, you don't have enough blood, so they will end up dying. So and at least to mitigate the severity, the vaccines are, at least now they are useful in these countries. Yeah, although it's not 100%.
Thank you. This also answers the second question of Jay Kishan about the malaria vaccine. And there was some other question from Dr. Braj Mohan. He's asking about whether it is possible to mutate the trunk of mosquito. And I think this type of questions regarding the vector, the change in vector, it's actually very difficult to do any genetic change in the vector, because and there were some studies actually where they released some genetically modified mosquitoes, which basically would be not producing any offsprings. They would be competing against the normal mosquitoes, but they would be sterile mosquitoes, so they would not be producing any offsprings. Those type of experiments require a lot of release of genetically modified mosquitoes in the environment. At a very large scale, which is actually very challenging. But does it actually? So those type of studies have been found to be promising. Brazil has done this, I think, in past.
Yeah, Brazil, and also some. It can be applicable to local, like a small pond.
Yes.
You can release them, because applying all over the generalizing, then it will affect the cycle, the habitat. The fish needs to feed the mosquito. The cycle will be affected, so it's very hard to get approval. So locally, the technologies are there, but you cannot generalize. Vector modification, there are a lot of techniques, but it's very, very hard to apply in a general, in the environment. Yeah, you affect the biodiversity. Yeah.
Okay. So thank you very much. I don't see any other question, and participants are thanking you for this amazing session. There's one
thank you very much.
question from Preeti. Can you find more variation in case of mosquitoes' innate immune signaling pathways, like you say toll or JAK state or other pathways genes? So the question is, can you find more variation in case of mosquitoes' innate immune signaling pathways, like JAK state or other pathways?
You mean in the mosquito, not in the mosquito? This study was in the humans.
Yes.
So
both pathways. Yes.
Once you have the list of genes that are differential, that are significantly associated with the disease, then you can take them, go to the pathway identifying in a KEGG or there are different softwares. Then you can identify the differences. Yeah.
Okay. So another question from.
Or you can if you are finding difficulty in associating, then you can do all genome sequencing. The whole genome sequencing will spit everything, all the variation, tiny variation. Yeah.
So another question from Ishwarya, the very interesting question is about, is there any study evidencing the role of mosquitoes microbiome in its vector potential?
Oh, that's a wonderful question. You should do PhD. That's true. So people now several labs, I know several labs in Yale and Johns Hopkins, they are using the microbiome within the insects.
Yes.
So to use as a like a delivery mechanism, like a paratransgenic technique, so they can modify those what's it called, the probiotics and the local microbiome to target the parasites. So they can secrete out the anti-parasitic, anti-parasitic, antimicrobial. So yeah, that's a very good technology. So you can modify, you can modulate the microbiome, the gut biome of the insect. Every insect, any living thing will have a bacteria. Okay, so that's a very good thing to modify. Use those as a weapon. Yeah.
So there were some studies showing the role of skin microbiome of the host. Human skin microbiome affects the biting preference of mosquito. So does it also affect a person's chances of getting malaria?
Definitely, Pushpendra. Yeah, those studies are ongoing, and because each skin microbiome is different between individuals as well. Yeah, exactly. Yeah, I agree. Yeah.
Okay.
There
So
could be a variation.
From Hariharan, interesting question that he is curious to know, why do mosquitoes circle over our head?
I'm not a vector biologist, but I'm sure there is some as Dr. Pushpendra Singh was telling, there's something based on the skin microbiome you get so different gases. I don't know. Or if someone knows, chip in, tell us if someone knows why they circulate on the head. Yeah, they're nasty actually. Yeah.
Yes. Okay, so if there are any no other questions, I thank both of our speakers for their time and their expert opinions, and for sharing their excellent work. And thanks to all the participants for their interest and these interesting questions. This was for one of the best sessions that we have had in this series, and we hope that we would keep improving, and we hope that this would be useful for other participants. And I think this much group of participants is good to deal with. I thank all of you, and we hope to stay in touch by email. You can send your questions if there are any, and we will pass on to the speakers, and you can note down their emails also from the Google profile that was shared with you in the flyer. And with that, if surgeon is there to give the formal vote of thanks, I request her, but I think she had some internet connectivity issue, so I would do that on her behalf, just to basically thank all of our participants and other members who have enabled in organizing this series. For example, Afjal and other members of our team who have created these links, these flyers. And thank you very much for all your time and interest. This encourages us to organize more such events later on also. Thank you very much to one and all. And again, thank you and for the Dashara festival also. And Dr. Senthil Rajagopal wants to say something. You can unmute yourself, sir. Yes.
Okay. Thank you so much for your kind invitation, Dr. Pushpendra Singh, and my co-speaker, Dr. Prakasha Kempaiah, for the excellent speech. I was very happy to see you as well as listen. I mean, learn your.
Yes, Senthil, same. Nice talk and background. I touched up on the application. That was easy.
Okay, thank you. Thank you, Pushpendra Singh. Thank you so much.
Okay, then.
And also our audience participant also. Thank you.
Thank you. And there is one answer about that this is related to the CO2 level that we exhale, that probably allows the mosquito to find us actually. Okay.
Yeah.
Bye.